This is Chapter 3, Part A. We're going to talk about the cell theory. The cell is the smallest structural and functional unit of any living organism, and the organism's functions depends on the functions of the individual cells, and the activities of cells are dictated by their subcellular structures. Continuity of life has a cellular basis. This means that cells only come from cells. There are over 200 different types of human cells, and they all vary in structure and function depending on what they do. As you see here, we have fibroblasts, erythrocytes, nerve cells, sperm cells, smooth and skeletal muscle epithelial cells, fat cells, macrophages, and many, many more. Cells have common structures and functions, and then they have individual types as well. All cells have a plasma membrane, which is the outer boundary, the cytoplasm, which is the intercellular fluid that contains the organelles, and then the nucleus, which is the control center of the cell. This is a generic cell. I've had several students be disappointed with no cell that looks exactly like this one. You see here, you have your cell nucleus, your nucleus, your nuclear envelope with pores. There's endoplasmic reticulum, both rough and smooth. Mitochondria, your cell membrane on the outside. It's part of your Golgi apparatus, and we'll go through these individually. The plasma membrane is going to take up most of the rest of this lecture because it is such an important part of the cell. Now, most people think about, oh, it's just a wrapper, but it is an intimate part of the cell's structure itself. We call it a fluid mosaic because it's sort of squishy and movable, and it definitely changes shape. Plays a dynamic role in cellular activity and keeps insides in and the outsides out. So separates intracellular fluid, that's insides, from the extracellular fluid, which is outsides. Interstitial fluid is also called extracellular fluid. It's the stuff outside the cell. Now, the membranes are made up of lipids, the phospholipid bilayer. We have the polar head, and they're polar and we also have glycolipids that make markers on the outside surface, as well as cholesterol, which is there for stability and fluidity. There's a lot of proteins in the cell membrane. Integral proteins are inserted into the membrane, and some go all the way through the membrane. We call that transmembrane. They function as ports or channels or enzymes or receptors. And then we have peripheral proteins, which are just on the periphery, the outside. They may be enzymes, motor molecules, linked from one cell to the other. And this is showing the general makeup of the cell membrane. Here you have integral proteins. This is a peripheral protein. On the outside, you have the glycocalyx. This has many functions, but part of it is the marker or name tag for that cell. Now, see, I've left out, oh yeah, the phospholipid bilayer. This part is polar hydrophilic, so the water outside the cell is not repelled by this layer. Here, this is hydrophilic. The inside is hydrophobic, and things that dissolve in water, as opposed to fats, can't get through here. So it's kind of like if this was a peanut butter sandwich. This is going to be the bread. This is going to be the bread. This is a peanut butter. Fat. Water can't get through it. Okay, functions of membrane proteins. Transport. Putting stuff in that needs to be in. So we have a channel here, and it's requiring ATP or energy to actively pump things into the cell. Receptors, such as for hormones, receptors for neurotransmitters. We also have attachments to the cytoskeleton on the inside and the extracellular matrix, so they hold the cell in place. Enzymatic activity. Many cells are going to have their surface with a lot of enzymes that increase the speed of chemical activity. Intracellular joining. This is cell-to-cell joining. Cell-to-cell recognition. If the cell recognizes this cell recognizes this cell, it's like a name tag on it. So if this is a cell from the immune system, and this is the one showing the proper name tag, then the immune system won't attack it. Okay, types of junctions. First we have the tight junctions. Very impermeable junctions here, so these layers here, holding the cells together. This might be things like in your skin, in your dermis, epidermis, to keep cells from, things from leaking in and out of the cell. Some of these are going to be reinforced by desmosomes. They're considered like a rivet type thing. They're made out of keratin and glycoproteins, and they hold that cell together. And then we have gap junctions. For instance, in the heart, each cell has to be able to communicate with other cells. So you have a channel that allows chemicals to go from one cell to the other. Membrane transport. Okay, the membranes are selectively permeable. Some molecules easily pass through the membrane, others do not. And we'll get into that further, but then types of membrane transport. Passive processes do not require energy or ATP. The substance just moves down its concentration gradient from an area of high concentration to low concentration. And then active transport requires ATP, and this can only occur in living membranes. Okay, we'll look at each of these individually. Okay, passive processes again. Diffusion is a movement of a substance from an area of high concentration to an area of low concentration. Now, if you look at these billiard balls here, they're held up by this rack. And they are in perpetual motion. And as you take this rack off, they start moving and they hit each other. As they hit each other, they bounce off. And so here we have high concentration, but as you move the rack, you'll have they'll spread out and then they'll be less concentrated. So they move from an area of high concentration to an area of low concentration. It's like this perfume in this bottle is concentrated into one little area, but when you spray it, it's released. And the chemicals bounce together and start to spread out where people nearby start to notice it. Okay, what determines if a substance can passively permeate a membrane. One is it lipid soluble? Because you remember you got that in-between area that's waterproof. So you got a lipid soluble substance such as oxygen. It goes right through. You also have to have channels of appropriate size. If a protein is too big, it can't get through. Some require carrier proteins to get through. Now, this is not a membrane channel. It's a doggy door. And this dog has no trouble getting through this door because it's just his size. This dog, however, unless he rips the door apart, can't get in or can't get out of the cell, whichever way. He might be a large anion, a large protein, and he can't get out simply because of the size that he is. Lipids just go right through the membrane. Things like anesthetics, fats, oxygen, anesthetics, alcohol, a lot of drugs just go right through membranes. Okay, facilitated diffusion require some type of carrier. Glucose is the absolute most commonly known one. It requires a carrier. You have to have insulin, and you have to have an insulin receptor. And these are very selective, and they're saturatable. And probably this accounts for a lot of your diabetes type 2. It's essentially Coca-Cola and candy bar poisoning. You have way too much saturation of these insulin and glucose carriers, the receptors for these, and they get saturated and eventually they burn out. Okay, and this is just showing, it's supposed to be lipid insoluble stuff. This could be a sugar. It really doesn't show the insulin though. Okay, now channels, protein channels. And these are going to be very important for ions. We're talking about chemical signals such as the signals that go down with muscle contraction and nerve impulses. Leakage channels are always open and they leak, kind of like my bathroom faucet. Gated channels are controlled by chemical or electrical signals. So they stay closed until something signals them to open. Okay, and here is a channel allowing stuff to go through it. Now, osmosis is the movement of water across a selectively permeable membrane from an area of high water concentration. That's water concentration to an area of low water concentration. So, as you have a cell here, and this is outside the cell. So this is a cell, and this is outside the cell. That might be the bloodstream. You're going to have little channels through here, and I'm not very good at drawing with this thing, but little pores. In the cell, if you have large chunks, they're trapped inside. And then you may have chunks here. Again, very difficult to draw with, and I always was not very good at drawing anyway. So if you have a whole lot of stuff, what stuff? Probably proteins, perhaps salts, outside the cell, this is very low water concentration. This is high water concentration. So water will diffuse this way. And what was a nice big cell here, trying to make it. Okay, and that's just what I was saying. Water concentration is determined by solute concentration. Osmolarity is a measure of the total concentration of solute particles. Does it matter what the solute is? Not really. Stuff that can't get through the membrane. It could be salt, it could be plasma proteins, whatever, it's just stuff. Okay, here we have this compartment has, let me get my hand back over here. This compartment has a lot of water, few particles. This one has a lot of particles. The particles are too big to get through these holes, so the water will pull here, and it actually will go against gravity to make this become higher. Water level to become higher. Osmosis enters or leaves the cell, and as if you change the volume of the cell, you disrupt the cellular function. Now, several terms we have here. Tonicity, the ability of a solution to cause a cell to shrink or swell. If a solution is isotonic, it has the same solute concentration as that of the cytosol, so water neither goes in or out. If it's hypertonic, the solution has a greater solute solution, too much, too strong, and so water's sucked out of the cell. Hypotonic solution, the solution has less solute, so stuff will go into the cell. Okay, now, is there anybody in here evil enough watching this ever poured salt on a slug? If you have, you know that it totally sucks the juice out of the slug. I was tricked into doing that at the age of 6, and it horrified me, and I was very remorseful. And I poured water on the slug, but he did not come back to life, I will add. There is such a thing as death. Okay, the reason that does that is because the salt collects on this mucous membrane of the slug, and since he's not waterproof, forms a hypertonic solution that draws fluid out of the slug, and it kills him. Now here we have, if you put a blood cell in an isotonic solution, normal saline like you inject people with would be a good example. You have neither water going in, water going out. If you put it in a hypertonic solution, you would not want to put the cell in 50% dextrose if you wanted it to actually live. It's going to shrink that cell. We call this crenation. If you put it in a hypotonic solution, the cell will swell. If you put cells in pure water and leave them there shortly, they will swell and actually rupture. Okay, this is a summary of passive processes. They all use the energy source, kinetic energy, because they're passive. They don't use energy from the cell. Simple diffusion is just movement of water from an area of high concentration, of substance from high concentration to low concentration. An example, movement of oxygen through the cell membrane, where you have a lot of oxygen outside it, not enough in it, such as around your lungs and the blood in your lungs, and the oxygen just diffuses across the membrane. Facilitated diffusion, movement of glucose in the cells. It has to be facilitated by insulin as well as a insulin receptor. Osmosis is movement of water through a selectively permeable membrane towards an area of high water concentration to an area of low water concentration, which also means towards a higher solute concentration. Okay, now we're going to talk about active transport. Now this uses ATP, only works in living cells because ATP, adenosine triphosphate. And as you recall from the last chapter, adenosine diphosphate takes on an extra phosphate, and that actually causes energy to be stored. And then when that phosphate is broken, energy is available for the cell to use. Two types we'll talk. Active transport require carrier proteins or solute pumps. They move solutes against a concentration gradient. And we have both primary and secondary active transport. Primary active transport, ATP, energy from ATP, causes transport protein change in the shape so that solutes are pumped across the membrane. Sodium, potassium pump, and we'll talk about this over and over again. It sits there and moves sodium out of a cell, potassium back in the cell to get it set up all for muscle contraction, nerve impulse. And secondary depends on an ion gradient created by primary transport. So it's basically energy stored in the ion gradients drives the transport of other solutes. Vesicular transport. A vesicle is a little packet of stuff, large particles, macromolecules, and it does require ATP. Okay, exocytosis, stuff going out of the cell. Endocytosis, stuff coming into the cell. Transcytosis, transport into, across, and then out of the cell, so all the way through. And then substance trafficking will get you 5 to 20. Oh, no, that's the different kind. Vesicular trafficking, transport from one area or organelle in the cell to another. Endocytosis and transcytosis often involve receptor mediated, therefore very selective, such as a neurotransmitter. Has to have a receptor or there's nothing for it to attach to. Phagocytosis is the engulfing, where the engulfs solid particles and brings them into the cell's interior. Macrophages and some white blood cells, such as neutrophils, do this. And this is what it looks like. It just basically has little receptors here. These are called pseudopods. The membrane moves forward. The the cytosol follows it. It comes around, closes, and just eats it like a little Pac-Man. And then generally it will either be used, destroyed, maybe by a lysosome may destroy it. Okay, endocytosis in the fluid phase is called penocytosis. And it is extracellular fluids are pulled into the interior of the cells. Perfect example, nutrient absorption in the small intestine. Many nutrients, even when we get them down to their smallest phase, won't just diffuse across the cell. So the cell has to take them in, physically eats them, and that closes up, forms a vesicle. Receptor-mediated endocytosis. You have a receptor here. This could be a neurotransmitter, for instance, that attaches here and goes into the cell. Or anyway, we'll go into the neurotransmitters later. Okay, exocytosis, spitting stuff out of the cells. This is an example. Here's the stuff in the cell you want to get rid of. It comes up here and attaches. It opens and then pulls wide and spits it out. Now I should probably go back to this one. Hormone secretion, neurotransmitter secretion, mucus, and getting rid of waste from the cell are generally by exocytosis, kind of like spitting out a watermelon seed. Okay, summary of active processes. All of them use ATP except secondary active transport. The molecules just follow an ion gradient. So primary active transport, pumping ions across the membrane, the sodium potassium pump. The exocytosis, secretion of hormones and neurotransmitters. Phagocytosis, white blood cells eating bacteria. Penocytosis, absorb cholesterol uptake. Hormone and cholesterol uptake. Okay, membrane potential. This is the separation of oppositely charged particles across a membrane. Now, we talked about that some elements tend to give away their outer membrane electron and therefore be positively charged. Others tend to accept one and therefore be negatively charged. So inside the cell, you have a lot of negatively charged ions, generally large proteins. And outside the cell, at rest, you tend to have a lot of positive stuff. So you end up getting a voltage across the membrane, and it can be measured. Now, maintaining that sodium-potassium pump continuously picks the sodium up, kicks it out of the cell, picks up some potassium, puts them back in the cells. And then the cells, potassium escapes, picks them back up, puts them in, throws a few sodiums out while it's at it. But the membrane interior becomes negative relative to the exterior because the large anions, proteins, they can't get out. Now, electrochemical gradient starts to attract potassium back into the cell. And then you finally get electrical gradient balances potassium concentration gradient. And because of this sodium, you get a little sodium diffusion into the cell, but generally when you get some, it's picked up and thrown back out. Here we have leakage channels, potassium going in and out of the cell. And we'll continue this discussion later on our next slideshow. I thought it'd be better to have them short so you wouldn't fall asleep until at least halfway through it. Thank you.